Okay, in this video we're looking at 17.7, which is autosomal linkage. We're going to possibly want our textbook and we're going to want our booklets open on page 3940 and maybe some scrap paper as well, just have a little think along. We're going to use this lesson to look at autosomal linkage plus a bit of time to get your head around some of the other topics from a few lessons ago. If you've got your textbook in front of you, there is an error on page 173 which I flagged up on your textbook error sheet. Can we just make sure that these have been corrected, please? Down here it shows you on this slide what they should look like. Little a, little b, little a, little b on both of those. Okay, so just before we start autosomal linkage, I'm going to cast your mind back to dihybrid crosses. Let's get rid of those. I'm going to get rid of this title so it doesn't confuse you, because I'm just looking at dihybrid for a second again. So let's imagine, because this will be a useful recap for us, that we've got two pairs of chromosomes. We know in our bodies we've got 23 pairs of chromosomes. The 23rd pair, in fact let's get this picture back, the 23rd pair are sex chromosomes, but all the other ones are what we call autosomes. In a dihybrid cross, we were looking at the inheritance of two pairs of genes on two separate pairs of chromosomes. So again, let's just have a little recap. So if we had these individuals, I've just made these genotypes up, in a dihybrid cross, I would lay it out like the parents were big G, big A, little a, crossed with another parent that I'm not really worried about at the moment. And when these guys came to make gametes during meiosis, you could have a gamete with big G, big A, or big G, little a, or little g, big A, or little g, little a. So you had four different types of gamete combination, didn't you? If this parent, let's say, was exactly the same genotype, they would make exactly the same gametes, wouldn't they? And then we would have our Punnett square. I'm just being a bit lazy here and not drawing everything out properly. And we had that massive Punnett square, and we had 16 possible combinations of offspring, ranging from big G, big A, down to little g, little a, little a. I don't suppose you can remember, can you, from a couple of lessons back, when we crossed two heterozygotes like that, what the expected ratio is. It was 9:3 to 3:1. That was quite a common ratio when you cross two heterozygotes together. So this would be your what, I don't know what these genes are coding for, but with all dominants, a bit of dominant, bit of recessive, and then the whole recessive ones. Okay, so if you've got two different genes, pairs on two different chromosomes, you end up with loads and loads of different combinations of offspring. What we're looking at today in autosomal linkage is when two genes are on the same autosome, the same chromosome. So this is dihybrid. Okay, you get a new piece of paper. For autosomal linkage, this time my two genes are on the same pair. Can you spot the difference there? Dihybrid cross, two different genes on two different pairs of chromosomes. Autosomal linkage, two different genes but on the same pair of homologous chromosomes. Now let's have a think about gametes. When this guy here came to make gametes in dihybrid crosses, we ended up with four different possible types of gamete, didn't we? When these go through meiosis, this big G and this big A, they stick together. There is no possible way that they can be separated. So your gametes will have either big G, big A in, or little g, little a, and that's the only possible combination. Now, to distinguish them from the same drawing here, we draw these a little bit differently. So rather than write just big G, big A like that, in the gametes, I draw the stick for the autosome and I put big G, big A like that. And the other gamete will have little g, little a in it like that. Okay, so it's just a slightly different way of writing things out. So we've got an example here in our textbook. Let's just see if it's in your notebook as well. Yeah, it is. So we've got in Drosophila melanogaster, this is a common species of fruit fly. The gene for body colour and wing shape is on the same chromosome. So dominant allele for grey body, little g allele for black body, capital N allele for normal shaped wings, and little n allele for vestigial wings. They're like little shriveled up wings. If you Google vestigial wings, you'll see what I mean. So here we've crossed a grey bodied fly with normal wings. So it's got big G, big N on one autosome and little g, little n on the other one. I'm telling you that. I mean, it could be the other way around, couldn't it? It could be big G, little n, little g, big N. But the question is telling you which goes with which. And we're crossing it with a grey-bodied fly with normal wings, again, with the same genotype. And we're going to write down what the gametes can be. So here we go. I think it's exactly the same example in your booklet here. So our parents, if we just copy straight out of the textbook, parents had big G and big N on one autosome, and on the other one little g and little n. And we're crossing them with another fruit fly with the same genotype. It doesn't really matter who's who, does it? But let's say this is the male and this is the female. Again, I've just been told this information in the question. The big G and the big N go together on this side, the little g and the little n go together on this side. Okay, so the gametes that they're going to make, we're going to have big G and big N in one gamete. I'm going to have little g and little n in another gamete. And we're crossing those with big G, big N gamete, and a little g, little n gamete. So just keep pausing if I'm going too fast. Now we're going to draw a Punnett square. But unlike a dihybrid cross where we had 4 different gametes for each side, we've only got 2. There's no possible way you can get a gamete with a big G and a little n from this set. These two are glued together. So we're going to draw our Punnett square out, but it only needs to be a nice 4x4, 2x2 grid. I'm going to move my gametes down here. Again, it doesn't really matter who's the male and who's the female, does it? And then we're going to join them together just like normal. So if this sperm and this egg join together, we're going to get big G, big N, big G, big M. We need space for the phenotype here actually, don't we? What's this going to look like? It's going to be a grey, normal winged fly. If these two gametes join together, we're going to get big G, big M, little g, little n, and that will be, again, because these are dominant, a grey and normal. If these two join together, big G, big N, little g, little n, that's going to be another grey normal. And if these two join together, I'm going to get little g, little n. My lines are getting longer, aren't they? little g little n, that's going to be a black vestigial wing. So in terms of my ratios here, I'm going to have 3:1 of grey normal to black vestigial winged flies. So if that had been a dihybrid cross and they were on different autosomes, the G's and the N's, I would have 4 different types of gametes for each side. I would have a massive Punnett square, and I would have had a 9:3 to 3:1 ratio. This time, because they're on the same autosome, I've ended up with only 2 types of fly, whereas in a dihybrid cross, I would have had 4 types of fly, phenotypically, 4 different phenotypes. Here I've only got 2 types of fly. So sometimes if you get a question and you can't work out if it's an autosomal linkage question or a dihybrid cross question, you can look at the outcomes you get. If you get fewer phenotypes than you would expect for a dihybrid cross, then it must be that they were on the same autosome to start with. I've put in the back of your book here some clues, because I know people were asking me the other day, how are we going to know when we get to these AQA questions which type of cross to do? You look for clues in the question. So if you see this clue here, the genes are carried on the same chromosome, that's telling you that it's autosomal linkage. Now, sometimes you can get really tiny deviations away from this. You know, like I said, there's not a chance that that big G could end up on the same chromosome as little n. Well, it's not technically true. There is a little way that that can happen. If you think back to one of your chapters last year, can you think what might cause this to happen? Let's go back to these chromosomes. So I start off with, let's just look at one individual, I guess. That's easier, isn't it? Let's take this male, and he's got big G, big N, little g, little n there. During meiosis, the first thing that's going to happen is the DNA will replicate. So you'll get 2 chromosomes that look like that, won't you? So you'll have a big G, big N, and it's replicated to big G, big N. And then this one, little g, little n, little m. And then during meiosis, these 2 line up on the equator of the cell, don't they? Do you remember where we get all of our homologous pairs lining up together like this? And then the spindle fibres come along and they separate those 2 out. That's the first stage of meiosis. Can you remember what sometimes happens when these homologous pairs line up on the equator of the cell? There's an unusual process can happen. If you remember, it's called crossing over. And so let's say these 2 bits here get so close they get tangled up. And that can result in a new combination of alleles. So you might get, let's draw these 2 chromosomes out again. So we'd get our big G, big N here. We'd get our big G, but let's say these 2 have been swapped over. Alright, so these guys then get pulled apart during meiosis. And then to form our gametes, these 2 get pulled apart and these 2 get pulled apart in the final stages of meiosis. So this time you might end up unusually with some gametes that have got these weird big G little n combinations in. So you wouldn't draw this out in a cross. I'll explain in a sec. So you would expect 99.9% of the time to have gametes that look like that in autosomal linkage. These ones are very unusual. And all I'm telling you about this for is if you get some results that you're pretty sure is caused by autosomal linkage, because you're getting a ratio of roughly 3 to 1 of these ones. But let's say you've had like 100 flies, 100 baby flies produced. You would expect 75 of the baby flies to be grey normal, 25 of the baby flies to be black vestigial. But let's say it doesn't happen like that, and you get this weird situation where you get like 74 of the baby flies were grey normal, but one of them ends up being grey with vestigial wings. And you're asked to suggest like what could have caused that. Well, that's going to be caused by crossing over. Very unusual process. In fact, I think there's this activity here called Tales of the Unexpected, where you've got roughly kind of a 3 to 1 ratio, but you've got these little weirdos here creeping in. That's likely to be caused by crossing over. There's nowhere in your booklet here to really draw out any notes about crossing over. I have put a note in here. Small deviations away from expected values are caused by crossing over, but there's no real way to draw that on a diagram. So feel free to add yourself in a little extra note somewhere about that. You've got a bit of space down here. Or you can go back to your textbook and draw out some diagrams and make yourself an extra little page there. That's fine. There's very few examples I can give you about autosomal linkage because it's not really come up and it wasn't in the old spec. So there's a couple of things I want you to have a go at. There's this mosquitoes question that came up. It's an old spec from a good while